Hello, my name is Kiya Govek, and I am presenting my poster for ISMB 2022 on a computational framework for the combined morphometric, transcriptomic, and physiological analysis of cells using metric geometry. Cell morphology reflects the progression of high-level cellular processes and is one of the most described cellular phenotypes in biology. However, computational methods that can summarize the great diversity of complex cell shapes found in tissues and infer associations with other single-cell data remain scarce. Here, we report on a computational framework named CAJAL for the morphometric and multimodal analysis of cells. CAJAL uses concepts from metric geometry to accurately build, visualize, and integrate cell morphology summary spaces and establish associations with molecular and physiological data of individual cells. The Gromov Wasserstein distance measures the minimum amount of deformation required to convert the shape of an object into that of another. We use this distance to define a summary space of cell morphologies. For each cell, we compute the pairwise distance matrix between points evenly sampled along its boundary. Then for each pair of cells, we compute the GW distance between their pointwise distance matrices using optimal transport. The resulting GW distance can be thought of as a distance in the latent space of cell morphologies. In this latent space, we can perform analyses such as visualizing a summary space of cell morphologies, using statistical and machine learning approaches to define cell populations based on their morphology, and inferring trajectories associated with the continuous morphological processes. We applied CAJAL to the dendrite reconstructions of neurons from the mouse visual cortex profiled with patch clamp. Cells with a similar morphology appeared in proximity in the UMAP representation of the space. The separation of excitatory and inhibitory neurons, cortical layer, and Cre driver line in this space also indicates the presence of morphological characteristics that are unique to each of these groups of cells. Lastly, the metric structure of the cell morphology space allows us to compute the middoid and average cell morphology to better visualize differences between morphological clusters. We quantitatively evaluated CAJAL compared to 5 other neuron morphometry methods and 4 other general cell morphometry methods using 3 metrics. We find that the GW distance is consistently the most effective at identifying morphological differences between Cre driver lines, transcriptomic types, and anatomical tissue origin. We used CAJAL to study the morphologies of over 70,000 cells from a cubic millimeter volume of the mouse visual cortex profiled from the Microns Consortium. This dataset includes not only neurons, but multiple types of glia and immune cells. Populations in the cell morphology space were consistent with 185 manually annotated cells provided by the Microns program. Neurons from different cortical layers were associated with distinct regions of the cell morphology space. And within the astrocyte population, it was apparent that layer 1 astrocytes were smaller, while layer 2-3 astrocytes were elongated perpendicularly to the plial surface. PatchSeq incorporates single-cell RNA-seq into whole-cell patch clamp, enabling concurrent high-throughput measurements of the transcriptome, physiology, and morphology of individual cells. We applied CAJAL to the dendrites of motor cortex neurons profiled with PatchSeq. The GW cell morphology space captured morphological differences between neurons from different transcriptomic types and cortical layers. We used the Laplacian score for feature selection to identify gene expression and electrophysiological programs associated with the morphological differences between neurons. Top gene ontologies associated with morphological differences in inhibitory neurons included cellular response to chemical stimulus, neuron differentiation, and taxis. The top gene ontologies for excitatory neurons included neuron projection morphogenesis. We also projected the RNA velocity field onto the GW cell morphology space to look for continuous transcriptomic changes that are also associated with changes in cell morphology. RNA velocity-related genes that were also associated with changes in cell morphology were enriched for genes involved in memory and cognition. We created a combined morphology space of full neuron reconstructions from these patch-seq datasets with neurons from the microns dataset. Manually annotated neuronal types from the microns dataset were closer to patch-seq cells of the matching T-type than to non-matching T-types, indicating that we integrated the technologies successfully. We also refined some of the manual annotations of the microns data with more precise transcriptomic definitions. For example, the microns Martinetti cell with a soma in layer 5 and a densely arborized axon projecting to layer 1 is closer morphologically to SST CRRNA2 T-type than the more general Martinetti SST CALB2 T-type. This matches previous results showing that layer 5 Martinetti cells that project to layer 1 express CHRNA2. Overall, we have shown that the application of metric geometry to cell morphometry supports the development of currently missing computational methods for integrating cell morphology data and modeling continuous morphological cellular processes. We expect that the development of these methods during the next few years will significantly impact our understanding of the relation between morphological, molecular, and physiological diversification of cells. I will be available to discuss this work at my poster during ISMB 2022, or also reachable via the email on screen.